Hey, welcome back to the series of lectures on methods. Let's start talking about some modern day methods now. We're going to get into basically everything but the neuroimaging in this group of slides. So, modern day methods are really all about mapping function to brain. Okay, so if you remember Phineas, we just talked about him in the last video. He had a frontal injury, and that resulted in some disruption of personality for a limited amount of time. I told you that popular culture made it seem like his personality was destroyed with his frontal injury. But really, you saw some personality changes right after the injury, for the small period of time right after the injury, and then there were some personality changes so when he started having seizures toward the end of his life. But really, it was not an overall complete personality change. And this really sparks this big argument between functional localists and non-localists, and thinking about the meaning of local. Like, what does it mean to be local? Are we talking about one area? Are we talking about a couple of areas that are sort of near each other? And the reason why I say this is because if you think about something like vision, right, you know that your eyes are involved in seeing the world, because when you cover your eyes, you can't see. Are your eyes completely responsible for vision? No. You can actually be blind at the level of the eye and still, and I'm doing finger air quotes here, you can still, quote, see. And it's something that we call blind sight, and we'll talk about that more in consciousness. But the eye and the brain are involved together in the whole process that we call visual perception or vision. And we'll talk more about that later. So let's start with our discussion on these different types of methods. And we'll start with lesions. The lesion method's been around for quite a while. It really got its start with Mr. Monk in 1884, and he was working with the occipital cortex of dogs, and what he found was something that he termed Seelenblindheit, and this is psychic blindness is what it translates to. And he found that when he lesioned certain parts of the occipital cortex in dogs, that they were able to navigate their world, they were able to avoid objects and not bump into things, but they weren't able to recognize objects. So they couldn't recognize a ball, they couldn't recognize their own bowl, they couldn't be trained to recognize any particular object, but they could still see in a particular sense. So this is, again, important to the whole localization of function argument. The next really big study when we talk about lesions, controlled lesions, is from Mishkin and Ungerleider. And this is a very, very famous study in the world of vision. This happened in the 1980s, and they were working with monkeys. And essentially what they found is that there are two separate pathways for vision, a what pathway and a where pathway. So let me give you a quick rundown of how they did this. So they had some monkeys, and they trained them on two different tasks. The first task, the monkeys were given one of two objects to learn, right? So they were trained, let's say, on this object here. Then they were presented with both objects, and the monkeys had to choose the novel object, the new one. Okay, so monkeys were trained to be able to do this task, and they were doing it successfully. They were also trained to do this task. These are two food bins here on the right and left. And the monkeys were trained to pick the food bin that was closest to the cylinder located on the board. Okay, so they were able to do this task as well. Now, when Mishkin and Ungerleider presented lesions to these areas purportedly involved in the where and what information, visual information, they found sort of like they found a double dissociation, right? So they lesioned the where pathway, and they find that monkeys are not able to do this task. They lesioned the what pathway, and they find that monkeys are not able to do this task. Okay, so this is a very important discovery in vision, but really, as far as the methodology is concerned, this is also very important when we talk about controlled lesions and the history of this particular methodology. So the next kind of lesion or controlled lesioning that we talk about is called transcranial magnetic stimulation, or TMS. And this is controlled lesioning, but really we refer to it more as temporary lesioning. So you use these nifty little figure 8 or infinity devices here, and they use an electromagnetic induction to induce electrical currents in a targeted brain region. So you pulse. You can either do single pulses, you can do multiple repetitive pulses in a particular brain area, and there's multiple reasons why we would want to use something like this, right? It causes temporary disruptions in neural firing, and it has quite a few uses, clinical uses. People have treated depression, they treat OCD with this. There are diagnostic uses, connectivity tests, right? So you want to test for brain damage, and where particular areas might be impaired. And then, of course, experimentally, there are numerous uses, many of them very entertaining. So let's have a look at this little demo.
I'm Roger Highfield. I'm the science editor of the Daily Telegraph. Now, words mean everything to me, but now I'm going to have the speech area of my brain turned off with a giant magnet. Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again. Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again. That may have looked like a strange party trick, but it's actually part of a serious research project conducted by Prof. Vince Walsh in a basement lab at the Institute of Cognitive Neuroscience in London. He's using electrical and magnetic brain stimulation to understand the most complex known object in the universe and how to treat conditions such as migraine and stroke. Just to show the power of the magnetic fields passing through my scalp, and that this wasn't a trick, this is what it did to our videotape, even though it was positioned a few meters away.
Star Trek. Yeah, it's very left eyebrow twitch.
It's hard to see because of the interference, but here I could still sing a nursery rhyme, even when he disabled my language centre, because singing depends on the other side of the brain. Okay. Humpty Dumpty sat on the wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again. I felt a bit apprehensive during this intriguing demonstration, but Professor Walsh thinks nothing of tinkering with his own brain for the sake of science.
Jack and Jill went up the hip. Yeah, that's great.
As researchers from around the world are about to gather.
Okay, so pretty humorous, but also very informative, right? So if we disrupt certain areas and we see disruptions in certain cognitive behaviors, in certain cognitions or certain behaviors, that's a great way to do research. Now, TMS can have some side effects. It can cause headaches, as well as treat migraines, right? So the kinds of stimulation that are going on in TMS we don't know exactly what's going on, right? We know that no one's ever really had a supremely adverse reaction to TMS, but we don't know exactly how we're stimulating an exact area of the person's brain. So moving on from the lesion stuff, we can talk briefly about direct brain stimulation. So before we were talking about controlled lesioning, and now we're going to talk about controlled stimulation. So this started with Wilder Penfield in the 1950s, and he was operating on patients with epilepsy and essentially found a map of the body in the parietal cortex. Now, how did he do that? He was using what we call the Montreal procedure, It allows patients to remain awake, and they can describe their reactions while a surgeon stimulates different areas of the brain. That sounds terrifying, but your brain doesn't feel pain. Like the brain, the organ itself doesn't feel pain. So once you get past your scalp and your skull, I can poke you all kinds of areas in your brain and you're not going to feel any of it. You're going to do all kinds of weird things because I'm stimulating different areas of your brain, but you're not going to feel the actual stimulation of the brain. So he was working around in patients' brains to essentially see where the problems were that were causing the seizures. And as he did, he discovered all these different kinds of areas. So what he found that's become really valuable in neuroscience is he was basically stimulating areas on the somatosensory cortex right here, his primary somatosensory cortex located in the parietal lobe. And he found that each part of the sensory and actually the motor cortex, which is right next to it, so this is primary somatosensory cortex, and right on the other side of this line is primary motor cortex and the frontal lobe. And he found that as he stimulated these different areas, that they actually corresponded to different areas of the body. So stimulating this area, for instance, patients would feel a sensation in their toe, or maybe their toe would wiggle, something similar, right? So you can see all of the different areas listed here that he found, using this controlled stimulation. And the body parts that correspond to them are drawn here in the form of what we call a homunculus. And the homunculus, basically, the size of the body part corresponds to the amount of cortex that's dedicated to that region. So we've got a lot of cortex dedicated to our face and sensation in our face because there are a lot of sensory nerve endings in the face, hands, feet. These are very important places for us to have, lots of cortex dedicated to. So that is controlled stimulation. Now we can talk about neurophysiology and electrophysiology. I realize now that neurophysiology has been misspelled on this slide this whole time, so I have fixed it, and hopefully I caught all the future places that it was misspelled as well. Sorry about that. Right, so let's have a look at a couple of the neurophysiological findings that really made a big impact in cognitive science. So this first guy over here looking all chill in his lab is Dr. Letfin. And he put out an article called What the Frog's Eye Tells the Frog's Brain. And essentially what he did was recorded from cells in the frog retina and realized that they were responding to small moving spots of light. And so this is kind of the first discovery of cells responding preferentially, or one of the first. So he kind of deemed them bug detectors, which was sort of the first proposition of something called a feature detector, which we'll talk a little bit more about later. But that is essentially, cells that are specialized for particular features in the environment. This paper of his is still one of the most cited papers in the Science Citation Index. So it's a very popular, very influential paper. The next big names in neurophysiology are going to be Hubel and Feisel in 1962, and they were doing recording from cells in cat occipital cortex. Yes, I know, I love kitties as well, but to their credit, we did get a lot of really good information out of these experiments that they did. And what they found, quite on accident actually, is that cells responded to oriented lines. The reason why I say on accident is because they were using one of these doohickeys right here. I don't know if you've ever seen one of these in action, but you have to load one slide at a time, right? So you've got a slide, it's projected, you got to remove the slide, pick another slide, put it back in. And so Hubel and Weissel were showing images, and what they found was that they weren't getting any responses from the neurons when the images were being projected, actively being projected, but they were getting responses as the slides were being removed from the projector. So what they found was that the neurons were actually responding to the edges of the slides as they were being pulled out of the projector. So that they would make a line across the screen as the slides came out. So they found a lot of really cool stuff with their experiments. They found a topographic representation of space in the visual cortex, which is essentially almost like a kind of like a picture copy of what's going on in your visual environment in the cortex, for lack of kind of a better description. They figured out cortical magnification, which is this idea that there are more neurons dedicated to the center of your visual field because there's a lot, you need to process a lot more detail in the center. You have a lot more photoreceptors in the center. We will talk about that a little bit more when we get into vision. So, from neurophysiology and electrophysiology, we will proceed to neuroanatomical stuff, and this is stuff like staining. So, staining and tracer dyes, it's pretty cool actually when these are done. PET, which we will talk about in functional neuroimaging, it uses tracer dyes in its imaging. Basically, you can do some really cool stuff with tracing. So you can figure out what cells are doing using stains. You can figure out how cells are made or how they're connected using stains. You can trace forward connections and backward connections. So, for example, here you've got this is the horseradish peroxidase method, right? So HRP is readily sort of absorbed by the terminals in the synapse here. So you put HRP in the synapse, then the cell will uptake some of that HRP and it will travel to here, and then you apply a staining technique and you can see where that HRP has traveled to within the cell, right? So it's pretty neat stuff that they can do with these stains, and we'll see some more staining when we talk about vision. So we'll be talking about Cajal and his staining of the retina. So, next time on methods, we'll be talking about functional neuroimaging. There are a whole lot of like little subcategories, sub approaches within functional neuroimaging. So it has its own lecture, and I will see you there.